Good afternoon, everybody, and welcome back to the Falling Walls Breakthrough Conversations. With this unique format, which is made possible in partnership with the Federal Ministry of Education and Research and Springer Nature, we hope to be able to take you with us into the minds and the experiences of our global science leaders, outstanding scientists, emerging innovators, and science entrepreneurs. This is your opportunity to engage in our discussions and to interact with our breakthrough winners and our guests, wherever you may be. I'm Angie Hassan, Associate Editor at Nature Communications, Springer Nature. It's an honor to welcome Anthony Hyman today at the Falling Walls Breakthrough Conversations. Anthony is a professor and director at the Max Planck Institute of Molecular Cell Biology and Genetics here in Germany, in Dresden. He is famous for his work on the role of phase separation in formation of biological compartments. He was awarded the Körber Prize for European Science this year. Congratulations.
Thank you.
For his work on research into cell droplets. And among being the recipient of several awards for his research, he is also a member of the German National Academy of Sciences, Leopoldina. He was also speaker this morning at the Falling Walls Circle plenary table dealing with the topic Mental Health for All, Global Action Plan on Dementia and Late Life Depression. And in our conversation, we will discuss more about his research and its implications in treating diseases. Welcome, Anthony.
Thank you.
Yeah. Phase separation is quite a complex process. Can you explain your research to the public?
Well, phase separation itself is not so complex. We see it every day in our lives when clouds form. You see, the water droplets forming a cloud by phase separation. If you make a vinaigrette with oil and vinegar, and it separates, you see the process of phase separation. So that's a well-known physical chemical phenomenon. Now you can go back into a cell and ask, how does it organize itself into a complex biochemistry? And I always like to set the scene by saying, let's just remember how complex we're dealing with, how the complex system. So a cell has 5 billion proteins, right, in one cell. It's about the population of the world.
Wow.
So every so often, a compartment will form to do something. And a cell is like a city. It's full of little factories that do things. But unlike the factories we make, they're very dynamic. By that I mean they're formed and they come apart when they're not needed. So it'd be like, okay, we need 100 Volkswagens, we'll build a factory, we'll build 100 and then it'll all disappear. I like to describe it more like a flash mob. You turn the music on, people will come together and they go away. Now, if you think about a typical compartment that might form inside a cell to do something, there may be 1 million proteins in there. So that could be the population of a city like Dresden or Berlin, actually. So in a way, you could think, okay, we've got 5 billion people in the world. There's 1 million people. They're going to come together to do something and they go away again, like, I don't know, Burning Man or something. It's complex, but it could work. But the cell is so different than that because if you scale up proteins to be people, but you maintain the same density as if they were proteins, we'd all fit in Lake Como. Right? We'd be tumbling past each other about 3 square kilometers a second and making the right interactions when necessary. Now, in that massively complex system, how is it the cells rapidly assemble and disassemble things? And our discovery was to show that phase separation is one of the ways that can happen. The same way that clouds form is a way that compartments can form, with the same type of physics.
And is this then implicated in disease?
Yes. So what we these compartments are we call liquids, and they're embedded in liquid cytoplasms. It's like you have a liquid drop floating around, and inside that stuff is happening. What we discovered in some diseases, these stop being so lively. The proteins inside them slow down, and they slowly stop, moving. And then it's something to do with the way the liquid transitions to more solid inside these drops that seem to be related to the onset of diseases like ALS. ALS is a form of neurodegeneration, like Stephen Hawking suffered from.
Right. And during the course of your research, what's one of the most surprising or exciting moments that you encountered?
Well, I think it was the moment we noticed that these small granules, we call them, pea granules, were liquids. As a scientist, it's a very hard and long-term business, punctuated by moments of immense excitement. So, it's probably, if you look at how long it took, well, maybe lead up to the discovery was, say, 10 years, and it's been 10 years since then, all of it extremely hard work. But then you have this moment that we had in front of us, monitor of a microscope, where we suddenly realized they're liquids. And then your heart starts racing and you start sweating and your palms get clammy. And you just have this moment where you think, oh, and then you understand that things are going to be different afterwards than they were before. And
that it was worth it.
It's worth the time and effort. Yes. Yeah, I think it is, because it's when I was a graduate student, someone told, there was somebody who worked with Sidney Brenner when they discovered the triplet code for DNA. And they went to the pub and they came back and looked at the plates, and it said it's a triplet code, right? That the proteins are specified by 3 base pairs. And this woman literally wanted to rush out and tell the world. And apparently Sidney said, let's stop and take a moment, because we are the only people in the whole history of humanity who've ever known that fact. And that's what I always tell them, let's stop and think, because we are the only people who know. It'll be a discovery for the world, but now we're the only ones.
Yeah, it's very exciting.
Yeah.
So as I mentioned in the introduction, you were awarded the Körber Prize for European Science. Could you tell us a little more about the specific research that won you this prize?
Yes, the specific research, I think, was the discovery of the formation of compartments by phase separation and their liquids. And I think more important, the understanding of how important it was. I think that was what was the right people at the right time. It wasn't just me, but it was a postdoc of mine at the time, Cliff Brangwijn, and Frank Jülicher, a colleague in theoretical physics. We were all together. And so we understood the implications at the same time.
Mm-hmm. And going back to mental health, so that has practical implications in terms of treating diseases such as Alzheimer's.
Right, exactly. So then the idea that these could change their material properties, that something, instead of being liquid, became hardened like a gel. It's like honey can be very runny, but if you leave it for an awful long time, it can get very hardened. Those transitions could be at the heart of some of these diseases, was very exciting, because it suggested treatments that if you stop that hardening, you could then sort of ameliorate disease.
And moving on to something a bit different now, you've worked in various countries. So what differences in the approach to science and the status of science have you been able to identify?
That's a very interesting question. I worked in UK, I worked in the United States, I worked in Germany. I mean, Germany is a wonderful place to work because it has this long-term commitment to funding science, basic science. I'm part of the Max Planck Society. I always like to say best job in the world. You're just given funding at the age of, how old I was, 36, for the rest of your career, to make basic discoveries. And so that is the great part of Germany. Now, the problem Germany has is accelerating. When you have a discovery, how do you accelerate, put more funding behind it? But in the United States, there's more difficulty in long-term basic funding. But Americans understand when it's an idea, let's pour time, money, resources into it, and they can suddenly accelerate. Yeah. So that's the sort of crucial difference, and UK sort of in between, I would say.
Interesting. Yeah. And as a successful scientist, and again, Körber Prize laureate, what advice would you give to young scientists who may feel frustrated at the progress of their research? So you mentioned that those exciting moments that make it all worth it. So what would you tell the young researchers who are still, at that stage where they haven't broken through their research and they're just working hard and nothing's working and it's super frustrating? Do you have any words of advice for them?
What I always say in that situation is, first of all, to be a good scientist, you have to be curious. You really have to want to know how the world works or something works. That has to drive you on. And then to remember that however frustrating it is, you have this unbelievable job where you're being paid by the taxpayer to come in every day and satisfy your curiosity. Most scientists on a day-to-day basis get very little direct instruction. They just do what they want for long periods of time. And there ain't no other job in the world like that. So I'd like to stand back and say, just enjoy the process and don't think about what's going to, the future, because that'll come on its own or not. There's no way to predict it because we're scientists, not engineers. But the process of doing it is a wonderful process. So just enjoy that process.
And if I could ask you personally, during your own moments of frustration in your own research, what are some of the things that have been able to inspire you?
I think I've always just been inspired by, reading a lot from successful other scientists about how they see the world. There's the Jacob Monod, the statue interior, the inside. He wrote about the discovery, what discovery meant, and just various people who've helped me understand what discovery really means. That's always been a great sort of help for me in those moments.
Mm-hmm. And in the plenary table earlier this afternoon, you mentioned celebrating the small wins. When it comes to mental health. Can you share with us where science thus far has won in this fight against Alzheimer's and dementia and mental disease in general?
I think, we have a good understanding in many cases of the genetic relationship, between gene modification, mutations, and disease. That's a big win because you know which proteins are involved, you know how the mutations, now you have to work out how you go from the mutation to the disease. But that's an incredible insight, especially in ALS. There's a lot of genetic relations. So I think that's been enormously important. I think there are very, very good mechanistic hypotheses. There's Alzheimer's, there's the amyloid cascade hypothesis, been around for a long time. There's our liquid to solid idea. There are a lot of good hypotheses. And what we're really just waiting for is, because the brain is so hard to access behind the blood-brain barrier, how do we get things in there that work? How do we test the ideas? So I think, there's a huge amount of basic information, but it's just about the issue of how we actually go about testing it.
And we know with mental diseases, for example, with Alzheimer's, it's a neurodegenerative disease where you just want to try to slow down the progress of the disease. Do you think there is any hope at all that we would ever actually reach a treatment, or is this just physiologically not possible?
I'm quite convinced that we will end up with a cure. Now, I'm 100% convinced in 100 years' time there won't be any neurodegeneration in the disease side. I mean, whether or not, to what extent we cure the aging part of it. All of us know that our grandmothers or grandfathers, when they were older, they may not have had degeneration, but brain didn't work quite as fast as when they were 20. Even we know our brains don't quite function like they did when we were younger. So that part of it is less clear. But the disease part, you get in your 60s, I'm certain that'll be solved as long as we accept that we have to do it through lots of little wins.
So very hopeful.
Chipping away at it step by step by step. Like I like to use the standard lifespan. I mentioned that earlier. We in Germany, I think men have a lifespan of 78. In 1950s, they had like 67. But it's every year a little bit better. Or HIV is a classic. When we first had some chemicals to help HIV, it wasn't very good. But now you basically keep the disease in check for most of people's lives. And those are young people. So I'm very hopeful.
Very good. I just want to remind the audience, if you have any questions, to just raise your hands. Do we have any questions from the audience? Okay, so I think we can continue. You have many responsibilities and commitments besides your actual research work. For example, I mentioned you're also a member of the German National Academy of Sciences, Leopoldina, right? So do you sometimes wish that you had more time to dedicate to your research, or do you enjoy doing different things?
I see life in very the sort of Buddhist tradition, the sort of 4 phases. You have the childhood phase, then the phase you're trying to make it, then the phase I'm in, and then the final phase. It's always been very helpful for me. And I had a great run doing research, for many decades, and money, and I could just get on with thinking about it. But eventually you have to transition to being more of a mentor, helping the next step, preparing yourself for the time after. There's no point working full time as a bench worker and suddenly 67, wham, you have to go off. So, so I feel very content with the more mentoring role that you take on and believe that's the right thing for senior scientists to do.
Very good. So maybe we can wrap up with our last question. Following up on that, what would you consider your favorite part of the job?
I think now, I mean, it changes, right? The favorite part of the job in the past was designing a good experiment and it giving you the result you wanted. It could either be a result that said it was true or untrue. That didn't matter, but it gave you a very clear result. So that used to be the wonderful thing about it, right? And now I think the favorite thing about my life is seeing young people be successful. You know, people leave your lab and they go off and run their own labs. That's a one, or they go off to industry, or they go off and do something else. They go into publishing, journalism, doesn't matter what. But they have come in, they've enjoyed the years in the lab, they've interacted, and they've gone on successfully to do something else. And that's always a pleasure.
That makes you feel proud.
Yeah, it does. It makes you feel proud, yeah.
That you somehow guided them with your mentorship and your supervision.
Yeah, you'd like to hope you've guided them a little bit, but at least you didn't mess them up. Maybe that's a more modest way to say it. But, you provide the environment, should we say, where they could prosper.
Mm-hmm. Yeah. Maybe we have time for just one more question. Is there any misconception about research in general or your research in specific that you feel the public may have, or about mental health in general that you would like you'd like to address?
The only thing, as I said on the roundtable this morning, is I think there's this kind of misconception because, generate a vaccine against COVID or develop antibiotics against bacteria that, cures happen overnight. Mm-hmm. That eventually someone with Alzheimer's will get a chemical, they'll be better. Whereas, but that probably will never happen. We will just, because they're different things. One is about correcting our own body, and the other one is about stopping invaders.
Right.
It's easier in a way to identify a foreign agent like a bacteria target and stop it than it is to keep our own bodies, healthy. So I think the main misconception is just to think, that scientists will one day work out with, I've got a cure. It's just to remember that even if you see a small win, is to celebrate it, mm-hmm. because that is the stepping stone to the next win and to the next win and the next win, until one day we know how to intervene early enough to prevent these dreadful diseases.
Right, celebrating the small wins.
Yes, the small wins, yeah.
Okay. Thank you very much for your time.
Thank you so much.
Very pleasant conversation. Thank you for our audience for tuning in. And now we will take a short break. So stay tuned on Zoom for our next conversation.